just to finish up this slide, you can see on the image on the right, which comes straight out of your textbook, at the top you have we're looking at from left, we're looking at the top left. It's a black and white image of a actual microscopic view of the nuclear envelope. And you have at least the visualization of the inner and outer membrane, as well as a nuclear pore. And then if you drop down to the black and white image that's at the bottom left, this is where you have this design of the nuclear pore. You actually have protein filaments that are arranged in a circular arrangement that have an opening in the middle with regard to, and that's what becomes the pore complex. Then moving over to the right in the artist rendition of the nucleus, you can see the nucleolus, the darker purple in the center, and then diffused throughout the nucleus is chromatin. That's the chromatin that will condense into chromosomes and then showing the nuclear envelope. You can see at the bottom right a black and white image of the nuclear lamina. What this is a mesh-like area that helps support the shape of the nuclear envelope. All of this is a topographical electron microscope. So remember it's that microscope that can actually pass electrons across the outer surface of something so that it can then visualize the texture of things. The nucleus houses DNA. DNA does not leave the nucleus, however, ribosomal RNA leaves the nucleus, as well as and via the ribosomal RNA is made in the nucleolus as well as the ribosomes, and they leave the nucleus to create proteins in the cell. And ribosomes here are made up of particles of ribosomal RNA and protein. So they tend to have small or two units, a large unit and a small unit. We call it a large subunit and a small subunit. You have a computer model on the left, which is kind of trying to show you how a ribosome works. Ribosomes are the site of protein synthesis, and these you can find ribosomes in two areas. They can be free floating in the cytoplasm. We call those free ribosomes, or they can be located on another organelle called the endoplasmic reticulum. And the endoplasmic reticulum, as well as the endoplasmic reticulum interfaces with the nuclear envelope. So we have them associated with an area where there is nuclear envelope and endoplasmic reticulum, but also on the endoplasmic reticulum itself. With regard to how a ribosome works, you have and we will go into this in some greater depth, but just to give you a little bit of a preview of how it works, it's almost like a vise that will hold the growing polypeptide together. So it's a vise that that allows for amino acids the time to form bonds, so that those bonds, and amino acids are the building blocks of proteins, of polypeptides, the monomer, if you will, and gives them time to form the bonds so that these chains can happen. And so with regard to ribosomes, generally free ribosomes make proteins that are used within the cell. So proteins and enzymes that are used within the cell. You can think of the cell itself needs to use an enzyme to break glucose apart to make energy. Those enzymes that are attached to the nuclear envelope via the interface between the endoplasmic reticulum and attached to the endoplasmic endoplasmic reticulum usually make vessel-type proteins, so proteins that are then going to be secreted outside the cell to be used elsewhere. You can think of a cell like the pancreatic cell that makes a very commonly known hormone called insulin. Insulin is a protein that's made by that cell and secreted out into the bloodstream so that it can be used by all cells of the body. Speaking of the endoplasmic reticulum, it is part of something called the endomembrane system, which we will talk about each of the organelles individually, but think of them as a continuous system of protein creating entities, but other things happen there also. But the biggest thing that generally gets talked about is protein creating entities. So areas of the cell. So endomembrane system. It consists of the nuclear envelope, endoplasmic reticulum, Golgi apparatus, vesicles, and lysosomes. The endoplasmic reticulum, as I said, we tend to abbreviate that as ER. And if it has ribosomes on it, it's considered rough ER. And it's the site of protein synthesis and processing. Processing meaning once a polypeptide is made, sometimes it is edited. So it becomes edited down, beginning, end, etc. The ribosomes are bound to the membrane, and the proteins that tend to be created by the endoplasmic reticulum are glycoproteins. So they're proteins that are bonded to carbohydrates. This is because these proteins, as I said, are going to be excreted by the cell, and being bound to carbohydrates allows those proteins, facilitates the fact that they're going to be turned into transport vesicles. And the endoplasmic reticulum also distributes these, the rough endoplasmic reticulum also distributes these vesicles and via the Golgi apparatus and surrounds them by membranes that then can be conjoined with the plasma membrane eventually. I think it's easier if we talk about it when you can actually see it. Smooth ER, which is that endoplasmic reticulum which lacks ribosomes, also is a site of synthesis, but what is made here are lipids. Think of the phospholipids that are needed to make more of the cell membrane, phospholipids that are needed to make any of the membranes. This is where you have metabolizing of carbohydrates. So metabolizing of carbohydrates, detoxifying poisons, and stores calcium. This is where you have the ability to take something that's potentially toxic to the body and make it water soluble by adding a hydroxy unit onto it generally. Also the site of stored calcium. Calcium is very, very important with regard to muscle contraction. So storing calcium so that it can be released in a timely fashion for a muscle contraction. Endoplasmic reticulum accounts for almost half of the total membrane in eukaryotic cells. And again, remember, it's continuous with the nuclear envelope. After the endoplasmic reticulum, whether it is the smooth, but more likely it's going to be the rough ER, the product that is created is then sent to the Golgi apparatus. And remember, this is all part of the membranous system. So this is in a certain fashion a continuous membrane, although the nature of it in each area changes. The Golgi apparatus is designed in these sacks that look like sacks of pancakes or pita bread, which are called cisternae. And that word gets used a couple of times. I guess it's, it's not that I guess, it's a word that describes an arrangement or a design. Cistern, flattened membranous sacs. What happens in the Golgi is it will receive the protein. So it receives the product of the ER. So whether that's rough or smooth ER, it receives those products, and those products are in vesicles, but that's not a finished design, I guess you might say. In very common parlance, we talk of the Golgi apparatus as the shipping and the receiving center, as it says at the top of your slide. So this is where the packages are sent. They aren't totally ready to be sent off yet. There'll be some tweaking to them, and also this is the area where they're looked at and said, okay, where does this go? Is this distributed inside the cell, or should I adjust this to send it outside the cell? And also you can think of it as this is where the decision is made, oh, this one should be expedited, this one shouldn't be expedited, in terms of how soon they should be exported from the cell. So processes them in any way that needs to be, they need to be processed, and distributes them to the place where they need to go. Certain macromolecules are manufactured in the Golgi apparatus, and as I said, sorts, packages, materials. The sacs are they're curved saccules, where you have an inside and an outside, or an inner and a trans. The inner cyst side is the receiving side. The outer trans side is the shipping side. And you can see that in this image where you have the cis face. So the cis or inner, I use that word loosely, inner face is the receiving, and the outer or trans face is the shipping. The trans face or the outer face shipping many times can actually turn into a vesicle itself. On the image in the lower left, you see a vesicle leaving the Golgi, and you can see where that actually looks like it came from the membrane of the Golgi, and that isn't far off the mark, where those the products that have been received from the ER have actually been sent to the Golgi. Golgi is deciding, okay, we're going to send this outside of the cell. We're going to put it in a special vesicle so that it can be sent out. It also works with lysosomes, so it's also involved in the secretion and lysosome formation. And lysosome itself is a organelle, membrane-bound organelle, but this is the Golgi apparatus is involved in the formation and secretion of lysosomes. Lysosomes tend to have enzymes in them or chemicals in them that can neutralize products that are potentially harmful to the cell. Speaking of lysosomes, here you have, they're produced by the Golgi apparatus, hydrolytic enzymes. So not only enzymes, but hydrolytic enzymes. And what happens is they have that hydrolytic, have the ability to break potentially harmful chemicals by digesting, I guess, by putting in water. So intercellular digestion. They can hydrolyze proteins, fats, polysaccharides. Polysaccharides, remember, are multiple monomers of sugary, or what you can think of carbohydrates, nucleic acids. It will fuse with a food vacuole and digest the molecules in that food vacuole. Also, lysosomes can use enzymes to recycle cells' own organelles. And we will pick this up on the next teaching portion.